Hello everyone and hello world. My name is Giulia and this is my final project for CS50's Introduction to Programming with Python. My project is called DNA Sequence Analyzer and the program simulates a simplified bioinformatics tool that analyzes a DNA sequence and returns several biologically relevant results. The user can input a DNA sequence either manually or by reading it from a FASTA file. In the project folder, I have provided a FASTA file for testing. The program, after the input, validates the sequence and performs multiple analyses, including nucleotide counting, GC content calculation, transcription into RNA, and generation of the reverse complement. Now I will briefly explain the structure of the project. This is the main file of the project, project.py. At the core of the program, there is a DNA sequence class, which represents a DNA sequence and contains all the analysis methods. The constructor, the init method, validates the sequence using a regular expression. It raises an error if the sequence is invalid. The method count nucleotides counts how many times each nucleotide appears in the sequence. The GC content method calculates the percentage of G and C nucleotides in the sequence, and the reverse complement function generates the complementary DNA strand in reverse order, and the transcription method converts the DNA sequence into RNA by replacing thymine with uracil. Finally, the print analysis method prints all the results in a readable format. Outside of the class, the main function controls the workflow of the program. It calls input method to allow the user to choose between manual input or input from a file. The get sequence function reads and cleans the user input, and the get sequence from file reads a FASTA file ignoring the header and the header lines and extracting the sequence. The second file is test project.py, which contains unit tests written using PyTest, and these tests verify that the main methods of the DNA sequence class work correctly, including GC content calculation, reverse complement, transcription, nucleotide counting, and invalid sequence handling. Now I will demonstrate how the program works. Okay, let me change the directory. Okay, so let's run the program. As expected, it prompts us to choose a input method for our DNA sequence. First, I choose the manual input by selecting 1. I then insert the sequence that I have previously copied, as indicated in the README file. And we can now see the output of the program. We have the sequence length in base pairs, the nucleotide counts. We do not have ambiguous nucleotides in this sequence, for example. And then we have the GC content percentage, the matching RNA sequence in which we can find U's instead of T's, and the reverse complement sequence. Now I will run the program again, choosing to input our sequence from a file. So I will select 2. The file name is test.fasta. I will insert the test, and this is the sequence of the keratin, for example, that is in my file. This is the sequence. We have the length in base pairs, the composition in nucleotides, the percentage of GC content, the RNA sequence, and the reverse complement. In conclusion, this project demonstrates how Python can be used to implement a simple but structured bioinformatics tool, combining object-oriented programming, file handling, and testing. Thank you for watching, and goodbye, world.